Welcome all. We are here today to propose our data science project titled Transcriptional Regulation of Spermatogenesis at Single Cell Level, which looks at analyzing and ratifying the role of transcription factors in the development of sperm cells. In our team, we have 4 completely qualified and competent data scientists: Rul Ayala, Lu Ni, Feng Hua, and William King Blaine. Male infertility is a prevalent issue in our society that affects more than 5% of men. Common treatment for male infertility cases is IVF, which costs on average $19,000 recorded by a 2013 study. By looking for transcription factors in certain cell populations, we may be able to identify genes that may affect male infertility. On top of that, this research could help with the reduction of the computational process in the analysis of gene expression. Now, a bit about spermatogenesis and genes. There are several lines of evidence that support that all living creatures contain DNA sequences called genes, and all of the cells within an organism contain the same genes. Then why is it that neurons in sperm, which have the same genes, have completely different functions? The answer lies in the way each cell expresses different sets of genes. In other words, the particular combination of genes that are turned on, expressed, or turned off, repressed, dictates cellular morphology, shape, and function. This gene expression is regulated by factors both extrinsic and intrinsic to the cell. Broadly speaking, we can say gene expression pattern is determined by information from both inside and outside the cell. For this to occur, there must be a mechanism to control when a gene is expressed to make RNA and protein, how much of the protein is made, and when it is time to stop making that protein because it is no longer needed. There are more than 50,000 genes within each cell of a mouse. Not all of them are able to make proteins. In fact, only around 40% of all genes are protein-coding genes. Within these protein-coding genes, there are very special types of genes that have a transcription regulatory activity, which are called transcription factors. Roughly 1,500 or 11% of these genes can produce proteins that help turn specific genes on or off by binding to nearby DNA. It is not the only way to control transcription, but sequence-specific transcription factors are considered the most important and diverse mechanisms of gene regulation.
Before we delve into this subject, let's talk about what is spermatogenesis. The tightly regulated process by which sperm is produced is called spermatogenesis. It begins with spermatogonial stem cells and results in the production of spermatozoa. All of these highly specialized cells are called germ cells. Many molecular events regulate the spatial and temporal differentiation of germ cells throughout all stages of spermatogenesis. Screening of candidate genes which are known to be expressed in some of these processes are included in diagnostic workup of male infertility. However, for the majority of infertility cases, including the most severe one, the underlying genetic cause is unknown. Detailed analysis of transcriptional regulation holds a key for understanding the cell biology process. Single-cell RNA sequencing is a relatively new technology that allows us to understand the cellular expression of genes. The first step in conducting the single-cell sequencing is effectively isolating viable single cell from tissue of interest. Next, we get read counts from the gene sequence of many cells. Finally, we can perform different analyses such as dimensional reduction and clustering. One of the most promising applications of scRNA sequencing is de novo discovery and annotation of cell types based on the transcription profiles. Here we hypothesize that the distribution of expression levels of transcription factors, the 11% of protein-coding genes, is sufficient for cell type identification in a single-cell RNA analysis. All the data used in this project is openly sourced from previously published gene sequences from male mice testes. We will combine the data from 3 previously unrelated data sets to do a clustering analysis. Here we have around 50,000 cells from Ernst, 3,000 cells from Law, and 401 cells from Steven at critical developmental time points. To start analyzing scRNA sequencing data and integrate all within the cell annotation data, we have some useful cell metadata, which is the most important data for this analysis is the read counts, which is a data frame where each row corresponds to a gene and each column corresponds to a single cell. The raw count values are not directly comparable between cells because in general, in this project, we use SC transform. The idea behind the SC
The next step is principal components analysis, PCA, is often performed to reduce the dimension of the data Prior to downstream analysis, it is the easiest way to overview the data is by transforming it and then visualize the first two principal components. In order to reduce the dimensions of our data, we use an alternative to PCA for visualizing single-cell RNA data called TSNE, the Distributed Stochastic Neighbor Embedding. It converts high-dimensional Euclidean distances between data points into conditional probabilities that represent similarities. The left graph uses k-means clustering to illustrate clustering of cells based on single-cell RNA expression. The right graph assigns cell types to these clusters based on which genes are expressed. The result of our clustering demonstrated a clear identification of a continuum of germ cell types in exactly the same order of the germ cell development. Our analysis using only 1505 genes had a similar outcome as the original data analysis using all 23,000 genes. In the same way, we have the same result using the Stephen dataset, which looks at testicular cells, whereas Ernst analyzes germ cells. Computationally, the annotation of cell types based on transcription profiles is a hard problem. Here, we demonstrate that the expression level of transcription factors are sufficient for cell type identification. It not only reduces the computational process by reducing the number of variables, it also highlights the importance of transcription factors in the control of cell identity and their association with male infertility.